Hello everyone. Today we are going to discuss about fluorescent microscopy. Myself, Jehrin J, MSc Biology, St. Mary's College, Thrissur, under the guidance of Dr. Kain Vadakan, Assistant Professor of St. Mary's College. Today we are going to discuss about fluorescence microscopy. Let's see in detail. What is fluorescent microscopy? A fluorescent microscopy is an optical microscope that uses fluorescence and phosphorescence in addition to reflection and absorption, to study the properties of organic or inorganic substances. Fluorescence is the emission of light by a substance that has absorbed light or other electromagnetic radiations, while phosphorescence is a specific type of photoluminescence related to fluorescence. In fluorescent microscope, the specimen itself acts as a light source. The specimen used to study are either fluorescent material or stained with fluorescent dyes. A chemical is said to be fluorescent if it is absorb light at one wavelength and emit light at a specific and longer wavelength. Most fluorescent dyes emit visible light, but some emit infrared light. Fluorochromosomes exhibit distinct excitation and emission spectra that depend upon their atomic structure and electron resonance properties. Two fluorescent dyes are commonly used are fluorescein, which emit an intense green fluorescence, which excited with blue light, and rhodamine, which emit a deep red fluorescence when excited with green-yellow light. Principles of fluorescent microscope. The most cellular are colorless and cannot be clearly distinguished under the microscope. The basic premises of fluorescence microscope is a stain, the components of dyes. The fluorescent dyes, also known as fluorophores or fluorochromosomes, are molecules that absorb excitation light at a given wavelength and after a short delay emit at a longer wavelength. The delay between the absorption and the emission is negligible, generally on the order of nanoseconds. The emission can be filtered from excitation light to reveal the location of the fluorophores. Fluorescent microscope uses much higher intensity light to the eliminate sample. This light excites fluorescent species in the sample, which then emit light of a longer wavelength. The image produced is based on the second light source or the emission wavelength of the fluorescent species rather than from the light originally used to illuminate and excite this sample. Now let's discuss about the design of electron microscope and fluorescent microscope. Typical components of fluorescent microscope consist of fluorescent dyes, the light source, the excitation filter. Fluorescent dyes, that is fluorophore. A fluorophore is a fluorescent chemical compound that can re-emit light upon light excitation. Fluorophores typically contain several combined aromatic groups or plain or cyclic molecules which have several pi bonds. Many fluorescent stains have been designed for a range of biological molecules. Some of these are small molecules that are intrinsically fluorescent and bind a biological molecule of interest. Major examples of these are nucleic acid strains like DAPI, host, forridine, which is used to stain actin fibers in mammalian cells. A light source. 4 types of light source are included. Xenon are lamps or mercury vapor lamps with an excitation filter, laser and high power LEDs. Laser are most commonly for complex fluorescent microscopy techniques, while xenon lamps and mercury lamps and LED with a dichroic excitation filter are commonly used for wide field epifluorescent microscopes. The excitation filter. The exciter is a band pass filter that passes only the wavelength absorbed by the fluorophore, thus minimizing the excitation of other source of fluorescence and blocking the excitation light in the fluorescent emission band. Dichroic mirror. The dichroic filter or thin film filter is very accurate color filter used to selectively pass light of a small range of colors while reflecting other colors. The emission filters. The emission is typically a band pass filter that pass only the wavelength emitted by the fluoropore and block all undesired light outside the band, especially the excitation light. By blocking unwanted excitation energy such as UV and IV or sample and system autofluorescence, optical filter ensure the darkest background. Working of fluorescent microscope. Light of the excitation wavelength is focused on the specimen through the objective lens. The fluorescence emitted by the specimen is focused on the detector by the objective. Since most of the excitation light is transmitted through the specimen, only reflected excitatory light reaches the objective together with the emitted light. Forms of fluorescence microscope. The fluorescent microscope refers to any microscope that uses fluorescence to generate an image, whether it is a small simple setup like epifluorescent microscope or more complicated designs such as confocal microscope, which uses optical section to get better resolution of the fluorescent image. Most fluorescent microscopes used are epifluorescent microscope, where excitation of fluoropore and detection of fluorescence are done through the same light path, that is through the objective. Uses of fluorescent microscope. To identify the structure in fixed and live biological samples. Fluorescent microscopy is a common tool for today's life science research because it allows the use of multicolor staining, labeling of structures within the cell and measurement of the physiological state of cells. Thank you.